Welcome viewers, welcome to the new lesson from Dr. R's Biology. If you did not subscribe the channel yet, you can just click on the subscribe button and bell icon. In this lesson, we are going to discuss about what is the exact or the basic difference between ABO blood group in human beings. So we will be discussing the facts behind different types of blood antigens. We know that human blood groups are mainly the following types that is represented in the table. That is starting from A positive and ending in AB negative, including O antigen or O blood group. So I'm just taking one example and discussing, and remaining thing you can just take from the table by pausing the video. So in case of the A positive blood group individual, the genotype of that particular individual can be IAIA or IAI0. Here, IAI0 is a heterozygous condition where I0 is actually recessive gene. And IA is a dominant gene that is responsible for the production of antigen A that you can see in the column that is represented for the antigen. This particular individual with A positive blood group is capable of producing an antibody against the B antigen, which will be normally present in individuals with the B blood group. Okay, in A positive individual, the rhesus factor or the Rh factor will be positive or it will be present in them. That's why it is known as the A positive blood group. I'm just taking one example only. Remaining you can just take from the table by pausing the video. If you pause the video and observe the table and studied what are the difference between different blood group individuals, just note down the difference that is observable among different blood group individuals. We can see that the main difference that is observable is nothing but the difference in antigen that is present on erythrocytes, or otherwise known as the red blood cells or RBC. So that is actually responsible for different blood groups in human beings. So the antigen that is expressed or that is present on the red blood cell is responsible for the different blood groups in human beings. So let us see what kind of antigen will be there on erythrocytes that is normally present in different blood group individuals. So you can see here, if you are just considering the first blood group, that is the O, why I am taking O is capable of producing no antigen, or otherwise known as the zero antigen. So let us see what kind of antigen is zero antigen, or that is present on the zero blood group or O blood group individual. So if you are just considering the O blood group, we can see that the RBC will be present, and on the surface of RBC, there will be a monosaccharide chain that is present. And this monosaccharide chain is nothing but this one. So you can see here the four main monosaccharides that is present, which is represented with the shape. So the first one, which is nothing but the yellow round structure, is representing the galactose monosaccharide. And this galactose is linked to the next monosaccharide through beta-1,3 glycosidic linkage, and the next monosaccharide, which is marked with the blue rectangle, is nothing but the N-Acetylglucosamine. This N-Acetylglucosamine is linked to another galactose sugar or the monosaccharide through another beta-1,3 linkage. And finally, the ultimate monosaccharide in this monosaccharide chain is nothing but the fucose, which is linked with the galactose, penultimate galactose, through alpha-1,2 glycosidic linkage. So this particular monosaccharide chain, which is composed of the four main monosaccharide, which is starting from the surface of the RBC, it is nothing but the galactose, N-acetylglucosamine, galactose and fucose, actually represents the O antigen, which is normally present on all kind of RBC among all different blood group individuals or human beings. Okay, so let us see the difference between O blood group and A blood group. So we are just considering the A blood group individual right now. So RBC is present, and on the surface of the RBC, same O antigen will be present. And the difference is there in the addition of one more monosaccharide to this monosaccharide chain. So there is a branch that is present in A antigen that is nothing but the N-Acetylgalactosamine that is attached to the penultimate galactose through alpha 1,3 glycosidic linkage. So if this N-Acetylgalactosamine branch is present in the monosaccharide chain that is present on the surface of RBC, the antigen will be A antigen. So this particular antigen that is represented there in the figure is actually the A antigen, which will be present in the RBC of A blood group individual. Now let us consider the B blood group individual and what is a B antigen. So in the same way, RBC is present and on the surface of RBC, O antigen will be present. Here, instead of N-Acetylgalactosamine branch, there will be a second galactose again, a third galactose unit again that is attached to the penultimate galactose, and that is with the same glycosidic bond, nothing but the alpha 1,3 glycosidic bond. So this particular galactose, if it is present, it will be B antigen. So the monosaccharide chain with the branch represented in the figure is actually representing the B antigen, which will be present on the RBC of B blood group individual. Now let us consider the AB blood group individuals. In case of the AB blood group, from the word itself we can see that there will be two antigen present on the RBC, and that is nothing but the A antigen and B antigen. So both A and B antigen will be expressed, and that's why this particular individual is capable of, or it is known as the AB blood group. Now you just recall the table that is presented in the early portion of this particular slides. So you can see that O blood group individual will be capable of producing antibody against A antigen as well as B antigen. So they will produce anti-A antibody and anti-B antibody. And the A blood group individual is capable of producing antibody against B antigen. That means they will be producing anti-B antibody. And the B blood group individual is capable of producing antibody against A antigen, that means the anti-A antibody. And the AB blood group, as they have both antigen present on their erythrocyte surface, will not be producing any antibody against A or B. So antibody will be zero in case of the AB blood group individual. So it is due to this fact O blood group individual is known as the universal donor, and AB blood group individual is known as the universal acceptor, because if they are taking or blood from any A individual, it will not be creating any problem because A antigen is already present in the individual, so antibody will be produced against only foreign antigens. So that is why it is known as O, known as the universal acceptor, and universal donor, and AB known as the universal acceptor. Now the question is, why there are different antigens, or what makes different antigens in different blood group individuals? The answer to the question is nothing but different antigens are there because there are different specific enzymes which are known as the glycosyl transferases that add extra monosaccharides to the O antigen. That means, for example, if you are just taking the type A individual will be having a specific type A glycosyl transferase, which is nothing but N-Acetylgalactosamine transferase that add N-Acetylgalactosamine to the penultimate galactose of O antigen. As a result, O antigen is forming the A antigen, or O antigen is converted into A antigen by the addition of N-Acetylgalactosamine. So you can just recall the figure that is in the previous slide. So you can see that A blood group individual is having an extra N-Acetylgalactosamine that is added. It is added by nothing but the enzyme N-Acetylgalactosamine transferase, or otherwise known as the type A galactosamine or type A glycosyl transferase. Similar is the case of B blood group individual, where they are having or their blood contains a particular type B glycosyl transferase enzyme, which will be adding galactose to the penultimate galactose unit. As a result, it will be converting O antigen into B antigen. And in case of the AB blood group individual, they will be having both type A glycosyl transferase enzyme and type B glycosyl transferase enzyme. As a result, they can add both N-Acetylgalactosamine and galactose to the penultimate galactose, forming both A antigen and B antigen. As we are going to find the basic difference between different blood group individuals, we have to ask the question again. How can different glycosyl transferase be there in different individuals? So what is actually making different glycosyl transferase? You know the answer, it is nothing but the gene that is inherited from the parent. So each individual that is capable of producing each type of glycosyl transferase are actually inheriting the gene responsible for the specific glycosyl transferase from their parents. And this gene is actually responsible for the different glycosyl transferase that is present in different blood group individuals. And also we have to keep in mind or remember the fact that both glycosyl transferase are almost identical except the position of 4 amino acid out of 354 amino acid that is present in the enzyme. So it is almost equal, and the difference is there in the position of amino acid only at 4 part. So what is the conclusion? So we can see that the individuals who are different in their blood group has different inherited genes, and there are mainly 3 genes, that is nothing but IA gene, IB gene and I0 gene, which is commonly known as the alleles. And we know that the blood group is actually an example for multiple allelism because more than 2 alleles are present in the same gene. So this gene IA, IB and I0 is actually responsible for the difference in different blood groups, where IA is capable of producing type A glycosyl transferase, which add N-Acetylgalactosamine to the primary antigen chain or O antigen to form or converting it into A antigen. And as a result, those individuals will be having A blood group. And the IB will be producing type B glycosyl transferase enzyme, which will add galactose to the primary antigen chain or O antigen and converting it into B antigen. As a result, the individual will be having the B blood group. I0 is a mutated allele or is capable of producing nothing, no specific type of glycosyl transferase, and as a result it is unable to add either N-Acetylgalactosamine or galactose to the primary antigen chain, and as a result the individual will be forming O blood group. Now you go back to the table that is present in the earlier slide, then you will understand this particular table in a well manner. So it will be clear for you, the table will be more clear for you after reading or after learning these particular statements. So finally, we can just say that the exact or the basic difference among the individuals with a different blood group is there in the gene inherited from the parent. After that, it is there in the ability to produce specific enzymes. Then it is there in the antigen that is expressed on the red blood cells. So the final conclusion is like this. If you are getting a question, what is the basic or exact difference among individuals with a different blood group, and getting choices like the gene inherited from the parent and specific glycosyl transferase enzymes present, then specific monosaccharide chain that is present on the RBC. So you have to first opt the gene inherited from the parent. If that is not there in the option, you have to choose specific enzymes. If it is not there, then only you have to choose the monosaccharide chain that is attached to the red blood cells, or otherwise known as the antigen that is expressed on the red blood cells. So the basic difference is gene, and the common expressible, or we can see the difference in the form of antigen that is attached. So that's the end of the lesson. Thank you for watching and hope it is helpful for you and valuable. So don't forget to subscribe Dr. Arts Biology. Thank you so much.